Okay, so now we are ready to get started with activity 5 and complete the procedure. Before we move on though, just a reminder, the objectives by the time we're done with the procedure here and answering the questions are that we can create a model of a Punnett square, and we're also going to be analyzing it. Okay, we're going to try to understand the information there to calculate the probability of specific traits being passed from parents to offspring. So 2 different things, creating and analyzing to determine probability. So we're going to begin by reading and summarizing the background section of how to use Punnett squares. Again, I'm going to model by reading the sections out loud, but I'm also going to be adding in some slides. Okay, I think it's really important for us to practice and get in the habit of reading our textbook and trying to make sense, the sense-making process on our own, but I'm here to help you as we go through. So we're going to read through and follow along on the slides as I set up a sample of a Punnett square. So it's a little bit blurry here, but you can always go in, and activity 5 will have the textbook link, will have this reading as well. So how to use Punnett squares, just a little bit of background. A Punnett square is a diagram that you can use to show the likelihood of each outcome of a breeding experiment. So we're talking about breeding, which means that we're talking about reproduction, sexual reproduction in this case. It is used when each parent's genes for a trait are known. We need to be able to know what those genes are. And there's a couple of different ways that this can be done. Technology helps us, as does past breeding experiments. We're going to fill in squares. By filling in the squares, you can find the possible genotypes and then the phenotypes of the offspring, and you can predict the chances that each phenotype will occur in the offspring. And so here's an example here of a Punnett square. We have 2 parents. One parent will be along the top and one parent will be along the side. Each parent has 2 possible alleles that it received from its own parents. Okay, and there's a chance that each one of these alleles can be passed on. So we have a spot for each of those alleles for parent 1 and a spot for each of parent 2's alleles. Okay. So now we're going to consider this cross between Sky and Poppy as an example. The 2 phenotypes for tail color are blue and orange. As you modeled with the colored discs and creature features, there are 2 versions, one for blue and one for orange, and these versions are called the alleles. As you saw when you did the coin tosses in activity 4 gene combo, the blue allele can be represented as an uppercase T and the orange as a lowercase. Now those letters, they can change. You can use B's if you prefer, big B for blue, for instance. But what doesn't change is capital versus lower. The uppercase letter always represents the dominant trait, okay? Whichever letter you decide to use, you need to use the same letter going throughout, okay? Just remember, uppercase is dominant. In this textbook, you are going to see that the underlined letter will be for the dominant, the capital, just to help distinguish them, because sometimes uppercase and or capitals and lowercase kind of look alike. Now, because Sky is from an island where there are no orange-tailed critters, no oranges at all, we can assume that only blue tail colors alleles are on that island. So he has to have a genotype that is big T, because there's only blue possibilities. Because Poppy is from an island where there are no blue-tailed critters, we can assume that she has an orange tail-colored allele. So her genotype is little t. Okay. Otherwise, we would have seen these other colors on each island. An organism that has only one kind of allele for a characteristic is called homozygous. Sky is homozygous for blue and Poppy's homozygous for orange. Okay, vocabulary, timeout. Okay, the prefix homo here, means the same letter, okay? We're going to compare that later on to something called heterozygous, but homozygous means the same. So it's either both capital, they're each the same, or they're each the same by being both lowercase. So here's an example of a Punnett square here, and this is the one that they use in your textbook, okay? And here you have Sky And here you have Poppy. And oftentimes, to help us keep it straight, you'll see like little gametes here. Notice that there's the little sperm because he's contributing the sperm. She's contributing the egg. So there's little eggs there. Okay. So we have our sperm contributor and our female. Okay. Notice that Sky was blue. So it's both. He only has blue. So each allele is a capital T. And then for Poppy, it's lowercase t. Then to fill this out, all you do is you drag the letters into each box going down and each box going across. So we're going to see more of that, and I will go through that more with you. Step 1, determine the genotypes. Step 2, label the genotypes along the sides. And then step 3, drag the letters down and fill in each box below and each box across with that letter. So that is how you do a Punnett square. The hardest part is remembering to get the parents on each side and figuring out what their genotype is. Once we do that, filling in the squares themselves, not so hard. So let's review. Now, this one is not in your book, but we're going to go through the same one. We're going to complete a Punnett square for what we call a monohybrid. That's a fancy word. Mono means one. A monohybrid, a one-trait Punnett square. And we're going to do this together. So the parents' alleles go on the outside of the square. Remember that. The parents go on the outside. So if we have Poppy, which is little t, by Sky, which is big T, big T. This X means that they're crossing, that it's a cross. It's like a time sign. So we're going to take Sky's up here, and we're going to put Sky, the dad, up on the top. It's okay if you put Sky on this side and then Poppy on the top. You'll come out with the same answer. But remember, he's big T. That means, and the second one is big T, so that means the second one will be big T. And going down, we have Poppy's little t and little t. Only one parent allele can go on the outside of each box. One from dad, one from mom. Now we're going to cross it. When I say cross it, what that means is that we are going to actually take the T, big T, and we're going to fill in anything in the column below it. So big T goes here and a big T goes here. And then the little t's will fill in across, horizontal. Okay, so drop the letters on the top into each square. So there's our big T. And notice the big T goes down again. And this big T goes down again. And now move each letter on the side into each square. So we have our little t. It's going across. See, it's going across that little t. And that goes there. And now we move this little t here and here. Okay. Now, there are 4 possibilities here. Okay, 4 possibilities, 4 squares. Keep that in mind. Now, the order does not matter when you drag them into the boxes, but the rule of thumb is that the capitals always go first. Now, if you put a capital second, I'm at the beginning here, I'm not going to be upset or anything like that because we're practicing. But good convention states that the capital is first. So now we've created, remember we're creating. Now we're going to analyze it and figure out what the heck that actually means. So if big T is the dominant blue tail color and little t is the recessive allele for orange, then we can use this Punnett square to make predictions. So what this shows us, if we analyze this, if we break it down, is that all of these are going to be blue. They all have a capital T in it. All the 4 boxes here have a capital T. So they're all 4. 4 out of 4 are going to be blue. That's the same as 100%. We can also write that as a ratio. Okay, where 4 boxes, 4 offspring are blue and 0, there are 0 possibilities out of these 4 boxes for orange. Yes, they have 1 orange allele, but remember that big T is dominant. That tells us about the phenotype, what we see. The genotype, the combination, we can't really see this as easily, is that they are 100%, all 4 out of 4 boxes are big T, little t. Or we can write it as a ratio. 4 boxes are big T, little t. 0 boxes are little t, little t. 0 boxes are big T, big T. So before we go on and we practice and you practice on your own on how to use it, why do we care about this? So what's a practical application? And many of us have dogs, or someone in our family has a dog, and we're looking at dog breeding as one of our podcasts. And dog breeding provides a great example of a practical way that Punnett squares can be used. The alleles of a particular species of dogs, they can be normal. In this case, we'll call that big D, or dwarf. Okay, so in this case, big D is normal. That means that the dominant trait is normal. Dwarf means small. If it's heterozygous, remember I told you before, homo meant same, hetero means different, big D, little d, and homozygous dominant, big D, these forms look the same because they each have a dominant allele in it. Both of them have a big D. They're both going to be tall. If you found a stray dog of this breed, could you determine its genotype? Now, if it was tall, could we know for sure if it was big D, little d, or big D, big D? Maybe not, because both of them are going to show a tall phenotype, so not necessarily. If it was little d, if it was a dwarf one, we might be able to. Okay, but at least it gives us a chance in the right direction. So could a dog be crossed with another dog to determine if he was a purebred for tallness? So in this last one, we can't tell if it's tall, if it's heterozygous or homozygous dominant, pure, only big Ds. Okay, it could be either or if it's tall, but we can do another cross to see if it is. So in order for us to do that, we have to figure out what type of dog we know 100% for sure what the genotype is by looking at it. And in this case, we're going to look at some different options here. If the dog is purebred, big D, it doesn't matter what you cross, all of the offspring will always look dominant. And you can tell because we'll have big D, 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 big D. If we are big D down here, let's see, we can draw D. I'm just dragging them down. Now I'm going to drag this one over. And this one over here. Notice they all have a big D in it though, so they're all going to be tall. And the same thing here, going down. In this case, going across. They all have a big D too, so we can't tell. If it's purebred, it doesn't matter what we cross it with, it's always going to be tall. Now, if we So let's try to think of a different one. So we know if it's purebred, it doesn't matter what we cross it with, but can we determine if it's a heterozygous, one of each? And that's what we're going to do here. So let me erase our, if I can do it, using screencast tools the Google Slides tools are a little bit more limited, the presentation tools. Okay. Now, if it's heterozygous, crossing it with a purebred is not going to help because obviously we just said it's going to be tall. But we can think here, what would the result be if you crossed it with another heterozygous? Would we be able to tell? So let's try it. If we had a big D, little d, and the one that we didn't know about happened to be heterozygous, We complete our Punnett squares. Big D's going down, little d's going down. Big D's going across, little d's going across. Okay, we should notice that we have 1 here, 1 out of the 4 that would be dwarf. Now, if we would do a homozygous recessive, one that was dwarf, that we could see for sure was short, we would know their allele combination here. And if we were to complete the Punnett square and fill them in, we have big D, big D, big D, little d, and little d, big D, and then another little d, 50% would be dwarf, and 50%, 2 out of 4, would be tall. And so the heterozygous cross would only give you a 20% chance of the recessive trait appearing. The most effective cross using the homozygous recessive. Recessive, I did not tell you, recessive is the opposite of dominant. The recessive is the one that does not show unless both alleles are recessive. So that's a practical application. We call that a test cross, and we're actually going to cover this more when we look at Mendel and his pea plants, and we're going to come back to these test crosses, okay? But the idea here is that hopefully you can see examples of Punnett squares being filled in, how we have the parents along the top and the side, and then we just drag the letters over and drag the letters down. Now it's going to be your turn. Okay, so at this point, we're going to stop the video. There's going to be a separate one walking you through how to do the student sheet, and you are going to do a Punnett square, and you are going to be writing in the letters, creating the Punnett square, and then analyzing it to determine the probability of traits being passed on. Good luck.